Hello everyone and welcome to today's drug target review webinar called Innovating Obesity Drug Discovery Trends, Challenges and Translational Strategies. My name is Sam Whitewick and I'll be conducting this webinar today. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for coming today. I'm happy to be joined by Dr. Luke Oostdyk to explore the evolving landscape of obesity drug discovery, highlighting key scientific trends, unmet needs, and translational challenges. Following the presentation, we'll move on to the Q&A session, so please submit your questions at any time during the discussion via the questions panel on the right-hand side of your screen. But before we start, we do have 3 poll questions that will show up on your screen in a moment. 2 will show up now, with a third coming shortly after the presentation. So the first question is, how familiar are you with the current approaches to obesity drug discovery? Very familiar, somewhat, or not yet? Or not familiar, apologies. That's how familiar are you with the current approaches to obesity drug discovery? Very familiar, somewhat familiar, or not familiar. And then we'll move on to the second poll question, which should be coming up in a moment. And that's it, is what stage are you in your drug discovery journey? Target validation, hit finding, hit lead, candidate selection, ND enabling, or other? That is what stage are you in your drug discovery journey? Target validation, hit finding, hit lead, candidate selection, ND enabling, or other? Thank you everyone for all your responses. And with that, I'll hand over to Dr. Oostdyk to begin the webinar. Over to you.
Hey everyone, thank you for joining today's webinar. We're excited you could join us. My name is Luke Oostdyk and I'm a study director from Eurofins Discovery. I'm here today to share with you some important trends, challenges, and translational strategies for the fast-growing field of obesity drug research. I'll start by sharing a bit more about myself. As I said, I'm Luke Oostdyk and I'm a study director for Lead Hunter in Vitro Discovery Services with Eurofins Discovery. I focus primarily on cell-based functional assays and have extensive experience in the development, design, and execution of these assays for the various stages of early drug discovery. Specifically for anti-obesity therapies, I've supported multiple programs for both small and large molecules. In the US, it typically takes 10 to 15 years and $1 billion to develop a drug. This journey of drug discovery and development from initial idea to regulatory approval is long, complex, and highly regulated, and each step is essential to ensure safety and efficacy. While it's displayed here as a linear process, many of these steps are often intertwined. Generally, we begin by identifying a biological target. This is typically a protein or receptor involved in a disease pathway. In the context of obesity in our talk today, this could be a GPCR like GLP-1R, for example, and this sets the foundation for therapeutic intervention. Next, compounds are screened to identify those that interact with the target. This can involve high throughput screening with binding functional assays or computational modeling to identify compounds of interest. These potential compounds of interest are screened to confirm they have the desired biological activity and taken through rounds of hit to lead and lead optimization using in vitro or in vivo studies to settle on clinical candidates based on potency, selectivity, and pharmacokinetic properties. Throughout this process, safety pharmacology testing in vitro or in vivo models are critical to rule out unmanageable toxicity or side effects. At this point, an investigational new drug, IND or similar application will be made, and upon approval, clinical trials will be initiated. Phase 1 focuses on safety and dosage in healthy volunteers. Phase 2 evaluates efficacy and side effects in patients. Phase 3 confirms effectiveness at scale and compares the drug to current standards. Now, if successful, a regulatory application is submitted for approval. Finally, phase 4 would monitor long-term safety and real-world performance after the drug hits the market. Today, we'll be talking about the ways that Eurofins discovery can support the discovery phases from target validation and hit finding to development candidate in obesity therapy programs. According to a recent study, greater than 14% of the world population is obese, and this number is expected to reach 25% by 2035. The economic burden this creates could be in the trillions of dollars. This need and the rise of GLP-1 receptor agonist therapies over the past few years have caused the obesity therapy market to explode, with the global market currently worth multiple billions of dollars. Despite this growth, or perhaps because of it, there are a variety of challenges that are currently faced in the weight loss drug discovery space. These include side effects such as nausea and vomiting with GLP-1 RAs that can limit adherence, and newer drugs aim to improve tolerability while maintaining or enhancing efficacy. Additionally, an estimated 10 to 30% of patients also do not respond to current therapies, and many do not start them at all due to the need for weekly injections, which highlights the need for personalized approaches, alternative mechanisms of action, or alternate modalities, such as small molecules instead of peptides. In the talk today, we'll be highlighting some in vitro approaches that can be used for improving efficacy, developing drugs for additional targets, and reducing side effects and safety liabilities. Longer term, there are issues with weight maintenance, undesirable muscle loss, access and affordability, and regulatory and reimbursement hurdles that also need to be addressed, but these are outside of the scope of what can be done in today's presentation. As I mentioned before, the main current class of obesity drugs are glucagon-like peptide 1 receptor, known as GLP-1R peptide agonists. These are typically based off the endogenous GLP-1 hormone with modifications to improve stability and half-life in the body. Single receptor therapies have achievable weight loss of up to around 15%. However, many have common GI side effects and do not work in all patients. We'll be sharing data with semaglutide, which is from Novo Nordisk, and it's known commercially as Wegovy for obesity in the talk today. GLP-1 RAs have also been developed that target additional receptors with the same drug for greater efficacy. Tirzepatide from Eli Lilly, which is known as Zepbound commercially, is a dual GLP-1R and gastric inhibitory polypeptide receptor agonist, which has enabled greater than 20% weight loss in patients. Another growing class of therapies are combination therapies, and these combine more than one drug to target multiple pathways. CagriSema is a combination therapy in trials from Novo Nordisk that combines semaglutide with the amylin agonist cagrilintide, and this dual pathway targeting was seen to be more effective at promoting weight loss than either therapy alone in clinical trials. Now, small molecule agonists make up another large proportion of the growing market. These typically have the benefit of an oral route of administration that does not require injections, which makes them both cheaper and enables better adherence from patients. We'll highlight data with orforglipron, a small molecule GLP-1R agonist from Eli Lilly, which is currently in process of seeking FDA approval for weight loss. In a recently published phase 3 trial, patients on the drug achieved weight loss of greater than 10%. Finally, other modalities such as antibodies are currently being pursued, and the assays discussed today can be used to develop these therapies as well. For the talk today, we're going to be sharing with you an example in vitro workflow for anti-obesity therapies using off-the-shelf services from Eurofins discovery. I'll highlight that a given drug program will have specific needs that may or may not be addressed here. And again, in the real world, discovery programs will not necessarily follow a linear path or proceed in the order presented, but we can help you design a screening cascade that is tailored to your specific program needs. If you look at the diagram on the left, the discovery stage starts with target validation and hit finding with the Obesity Light panel. This is a focused set of assays specifically for receptors that play a role in metabolic processes that control weight. Following this is potency determination for active targets in multiple assay formats, in-depth mechanism of action studies to quantify things like bias signaling, and finally specificity testing with panels like GPCR Max. We then progress to the development and preclinical validation phases with in vitro safety pharmacology testing and biological modeling. Finally, though I won't be sharing this data in today's talk, candidate molecules can be tested for efficacy and safety using translationally relevant in vivo obesity models. Relatively recent advances have revealed many of the molecular mechanisms between the gut and brain that control appetite and eating behavior. Various gut-brain peptide hormones are secreted in response to nutrients and other factors, which include leptin, insulin, glucagon, amylin, peptide YY, glucagon-like peptide 1, gastric inhibitory polypeptide, and orexin, among others, that then travel through the circulatory system to the brain to signal through their cognate receptors. These are primarily G protein coupled receptors, and they elicit additional signaling through neurotransmitters such as melanocortin and neuropeptide Y, which ultimately control the body's appetite and caloric intake. Dysregulation of these pathways can result in metabolic dysfunction, leading to diseases like obesity, which make them key pathways for potential therapeutic intervention. We took this signaling axis and assembled a fit-for-purpose collection of cell-based assays we call the Obesity Light Panel. The first step in our sample workflow, this panel can be used for target validation or focused hit finding for anti-obesity therapies and consists of 25 assays that measure activity against 13 receptors from the glucagon, amylin, cholecystokinin, ghrelin, leptin, melanocortin, orexin, and peptide YY families. If you look at the table on the left of the slide, you can see that each receptor is profiled in the assay mode, either agonist or antagonist, that is most relevant for anti-obesity therapies, and the panel includes both beta-arrestin and second messenger assays for GPCRs. The cell-based assays in this panel utilize a technology called enzyme fragment complementation, or EFC. EFC technology consists of a split beta-galactosidase enzyme with two components, an enzyme donor and an enzyme acceptor. These fragments are inactive separately, but when brought in close proximity, will complement to form an active beta-gal enzyme. The two fragments can be tagged to proteins of interest to interrogate biological events in a cell, with addition of a luminescent substrate resulting in generation of light that can be measured on a plate reader. This technology has been used to interrogate a multitude of cellular responses in proteins, including G protein coupled receptors, kinases, interleukins, nuclear hormone receptors, and epigenetic and other cellular pathways. They're suitable for use with many different modalities, including small molecules, peptides, large biologics, or ADCs, and I'll share data with both peptides and small molecules in the coming slides. We profiled three leading GLP-1RA peptides, semaglutide, tirzepatide, and retatrutide, in the obesity light panel at 1 micromolar to validate their targeted activity. Data shown is normalized to a maximum response of a reference compound for a given assay. Well, I'm sure many of you are familiar with these drugs. I'll give some background on each as I walk through the data and start with semaglutide from Novo Nordisk. As I mentioned previously, it is a peptide based off the endogenous GLP-1 sequence and has select amino acid changes shown in the figure at left, which include a modified lysine residue that is lipidated and an alpha amino isobutyric acid residue, and both of these help to prolong half-life and increase stability of the compound in the body. It has been approved since 2021 for obesity and showed weight loss up to 15% in phase 3 trials. In the obesity light panel data on the top right of the slide, semaglutide has shown specific activity for GLP-1R in both arrestin and second messenger formats, and it is a full agonist in both assays. Next, we have tirzepatide, which is from Eli Lilly. It's a dual GLP-1 GIP receptor agonist. And if you look at the sequence on the left of the slide, you can see that it's based on the endogenous GIP sequence, and it has modifications to impart activity against GLP-1R and promote stability in the body as well. With the addition of GIP agonism, tirzepatide treatment resulted in weight loss of around 20% in phase 3 trial, and is approved for weight loss under the name Zepbound. In the middle panel on the right, tirzepatide is fully active in second messenger assays for GIP and GLP-1R. In the arrestin assay, it is a full agonist for GIP, but only a partial agonist for GLP-1R. And this is something that we'll investigate further in later steps in our workflow. Finally, we have retatrutide. This peptide, which is also from Eli Lilly, is now a triple GLP-1 GIP glucagon receptor agonist. And it's also based on the endogenous GIP, but it now has additional substitutions to have both GLP-1R and GCGR agonism. The drug is still in clinical trials for both obesity and diabetes, but it has shown promise of more rapid and substantial weight loss of up to 25% in a phase 2 trial. In the bottom panel now on the right of the slide, retatrutide is a full agonist in second messenger assays and a partial agonist in the arrestin assays for each of these three receptors. We'll discuss the benefits of arrestin for identifying partial agonism in a few slides. One of the trends I talked about before was the advancement of small molecule agonists to address some of the challenges faced with those peptide agonists. which include cost and adherence in patients. Orforglipron is a non-peptide small molecule agonist that is currently in clinical trials and recently demonstrated about 12.5% average weight reduction in a phase 3 trial. Eli Lilly is expected to apply for approval as a weight loss therapy with this drug by the end of the year. With the obesity light panel data now at the bottom right of the slide, we see that orforglipron is highly specific in this panel and actually only active against GLP-1R in the second messenger assay, but has no activity at all in the beta-arrestin format. Overall, the obesity light panel profiling quickly and accurately evaluates activity of peptides, small molecules, biologics, and other modalities for select targets relevant to obesity research, and it serves as a starting point for further studies to determine potency and investigate mechanism of action. This leads us to the next step in our sample workflow, which is potency determination in multiple assay formats for those active targets from the panel. We'll first look at beta-arrestin signaling. Now, GPCRs have two main signaling pathways, beta-arrestin and G-protein recruitment. G-protein signaling activates second messenger pathways like cyclic AMP or IP3 to trigger rapid cellular responses, while beta-arrestin signaling typically regulates receptor desensitization and internalization, and it can also initiate distinct slower signaling cascades. The beta-arrestin recruitment assay measures direct activation of a receptor using EFC technology with a tagged receptor and arrestin as shown in the diagram on the slide. One benefit of this system is high specificity of response due to the dual tagging. Now, generally, arrestin assays are not the most sensitive, but they are very useful for identifying partial agonism and measuring signaling bias, which we'll talk about later. It's also an assay that is easily scalable for high throughput screening. If you look at the data on the top right of the slide, we have dose response curves for GLP-1, GIP, and GCG for the three peptides of interest, and reference compounds as well for each assay, which show nanomolar potencies for each of our peptide agonists for their respective targets. And this also clearly illustrates the partial agonism of tirzepatide and retatrutide at GLP-1 for this pathway. The second GPCR signaling pathway that we'll talk about is G-protein signaling. Upon activation with an agonist, receptor-specific G-alpha proteins are recruited that initiate distinct downstream signaling cascades. GS and GI proteins increase or decrease intracellular cyclic AMP levels respectively via adenylate cyclase, while GQ activates phospholipase C to ultimately release intracellular calcium. The GPCRs of interest in our talk here, GLP-1, GIP, and glucagon, are all GS coupled, and these increase cyclic AMP levels. For measuring these levels, we again use EFC technology in a competition assay as seen in the diagram on the slide. Due to the signal amplification of the second messenger cascade, these assays are generally more sensitive than arrestin recruitment, which can be beneficial for structure-activity relationship studies. This signaling amplification does mean there's typically less ability to discern partial agonists, and the sensitivity comes with the slight trade-off of higher potential for non-specific activity due to these measurements being downstream of actual receptor activation. The data on the top of the slide now for the three peptides illustrates these points. Each peptide has picomolar potency for its given receptor and are also all full agonists compared to our reference control. A third way to evaluate compound potency is by direct compound binding in a radioligand competition binding assay. This assay measures the compound's affinity and potential to displace endogenous or therapeutic ligands and provides key insights into target engagement and selectivity. The assay has the benefit of being independent of any intracellular downstream signaling pathways or receptor expression levels. This means it could permit quantitative comparisons of compound activity across our different receptors. In the binding data at the top of the slide, we can compare retatrutide, for example, which binds in the nanomolar range at GLP-1 and GIP, versus the micromolar range for the glucagon receptor. This allows decoupling of compound binding from the functional responses measured in the other two assays. Together, these complementary assay readouts offer a multidimensional view of compound activity. They cover a wide range of potencies from the micromolar to the picomolar range, and collectively they support target validation, lead optimization, and help provide mechanistic insights. When plotted on the same x-axis as shown on the slide now, relative potencies of compounds in the different assay formats are easily apparent. Now, this is a starting point to inform detailed characteristics of compounds such as biased agonism, which we'll be discussing next as we progress in our sample workflow. In biased GPCR signaling, a specific ligand may signal through either beta-arrestin recruitment or G-protein signaling preferentially. As shown here in the diagram at right, while a biased agonist signals equally through each pathway, a beta-arrestin biased agonist signals more strongly through the arrestin pathway, and a G-protein biased ligand signals more strongly through the G-protein pathway. Biased agonism toward G-proteins and away from beta-arrestin has been proposed and demonstrated to be therapeutically beneficial for receptor classes such as opioids, where the adverse effects such as respiratory depression, constipation, tolerance issues, and addiction have been more strongly linked to beta-arrestin signaling. For GLP-1, GIP, and other obesity-related receptors such as melanocortin-4 and ghrelin, various publications have indicated a benefit of biased signaling. This includes anti-diabetic effects and protections against obesity, which makes bias an intriguing prospect for therapies. For GLP-1R, there's growing evidence that the second messenger pathways are responsible for the appetite suppression, insulin secretion, and glucose lowering that's seen, while signaling through beta-arrestin can lead to desensitization, receptor internalization, other things that attenuate the beneficial activity of receptor activation. This beta-arrestin signaling may also potentially promote the observed side effects like nausea. For these reasons, there's great interest in development of G-protein biased agonists. Now, because of the inherent differences between arrestin and second messenger assays, comparisons of potency measurements between the two cannot be used to evaluate bias directly. Dr. Terry Kanakin has published a model that allows for comparing responses between different pathways by deriving a transduction coefficient that utilizes the ligand and pathway-specific parameter tau and the ligand receptor-specific parameter Ka. This talk isn't going to go into detail on the underlying math, but these parameters are used to calculate a log bias value that compares responses between the two pathways for a compound relative to an unbiased reference compound. On the left of the slide now, we have data for semaglutide, tirzepatide, and retatrutide comparing signaling bias between arrestin and cyclic AMP at GLP-1R compared to the neutral reference exentin-4. This data is now plotted slightly differently than the previous data we have shown because bias calculations require normalizing to the maximum possible response in a given assay system. We use force length stimulation in the cyclic AMP format and an EFC control in the arrestin format to determine the maximum possible signal in each assay. Additionally, one curve for each assay is shown here, but the GPCR bias platform utilizes multiple replicates to enable statistical significance. Visually now in the response curves, you can see there are differences in compound response for both potency and efficacy between the arrestin and second messenger assay. Moving now to the graph on the right, this shows us our calculated potency bias on the x-axis and the relative intrinsic activity, which is related to efficacy of the response on the y-axis. Absolute values greater than 0.5 are generally considered to be significant, and this data shows that we measure slight but minimal bias for these three compounds, but a strong efficacy imbalance with tirzepatide, which is reflective of the minimal efficacy it shows in the arrestin assay. Now, this is in agreement with published data from Eli Lilly for tirzepatide, which demonstrated it is an imbalanced agonist, and this could be part of the reason it shows increased efficacy in patients. To delve deeper into the signaling intricacies of each peptide, we can also measure bias relative to arrestin recruitment in additional signaling pathways that measure downstream or alternate signaling cascades, which includes receptor internalization. Starting now with the diagram in the bottom center of the slide, we have activated internalization. This measures beta-arrestin dependent receptor internalization with EFC fragments on beta-arrestin and the endosome respectively. If you look at the graph above this, you can see the response for each compound is similar to the beta-arrestin data we have shown, where we have partial agonism apparent for tirzepatide and retatrutide, and potencies are in the nanomolar range. Moving now to the right, we have total internalization. This measures all forms of internalization as the GPCR itself is tagged instead of beta-arrestin in addition to that tag on the endosome. Interestingly, in the graph above, each compound demonstrates now greater full agonist activity, which suggests that beta-arrestin independent forms of internalization may play a role with these compounds. Finally, on the right, we have beta-arrestin 1 signaling. This assay format is identical to the beta-arrestin assay we have shown so far, but it instead utilizes the alternate isoform beta-arrestin 1, which can initiate alternate signaling cascades instead of the canonical beta-arrestin 2 pathway. For these peptides, the different arrestin isoforms have similar potencies and efficacy. And if you look now to the graphs on the left side of the slide, we've plotted bias and intrinsic efficacy for these three assays, along with the cyclic AMP data from the previous slide, all relative to the canonical beta-arrestin 2 pathway. We did not detect any significant signaling biases in the comparison of beta-arrestin 2 in these assays. And the only significant signaling difference is again the efficacy imbalance with tirzepatide in the cyclic AMP assay. Overall, this platform supports targeted development of compounds with very specific pharmacological properties. Continuing with our MOA studies, we'll look at some orthologous assays. Prior to moving a program from in vitro profiling to in vivo experiments, it can be important to confirm the activity seen in human receptors is consistent with orthologous species where in vivo experiments might be performed. Now, due to differences in relative homologies between the receptors of different species, such as mouse, rat, or cynomolgus macaque, there can be differences in specificity and relative potencies of compounds for each orthologous receptor. The data on the slide now shows second messenger cyclic AMP modulation of GLP-1, GCG, and GIP receptors, with data for the triple peptide agonist retatrutide and the single GLP-1 agonist and small molecule orforglipron with the human, cyno, rat, and mouse receptors. Retatrutide is active for all 4 species, and you can compare relative potencies between the 2 assays for given species. For example, we see that it's more potent than GIP at the human GIPR, but it's actually less potent comparatively at the mouse GIPR. Now, orforglipron, on the other hand, is highly specific for human and cyno GLP-1R, and it's nearly inactive at the mouse and rat receptors, as you can see on the slide now, where we have just GLP-1R data plotted. This has actually been published and was determined to be due to a single amino acid change at tryptophan 33 between the rodent and higher mammalian receptors, which is critical for the drug's binding. Identifying this species specificity was crucial in that study and meant the authors did not perform any experiments with rodents and instead used the cynomolgus macaque for pharmacokinetic and other in vivo experiments. This really strongly illustrates the utility of having orthologous assays for a variety of species early on in the discovery process. The final step of our sample workflow is broad specificity testing. Since we've now confirmed activity, potency, and looked at in-depth mechanism of action, we can progress to look at this specificity. For this, we use the GPCR Max panel, which includes 168 Class A and Class B GPCRs, assayed for arrest and recruitment in agonist and antagonist modes, typically at a single concentration, but it can also be tested in multiple concentrations if desired. On the slide now is a phylogenetic tree of GPCRs in the panel with the data for 3 anti-obesity peptides presented as circles representing the magnitude of their response surrounding the tree, either for agonist mode on the left or antagonist mode on the right. We saw no off-target activity observed, including at the other most closely related Class B receptors, which you can see in that insert at the bottom of the slide. The GPCR Max panel is a great tool for confirming compound specificity and also identifying unwanted off-target effects. In alternate workflows, this panel could be used earlier in the cascade, even for target identification as well. Additional panels are also available for other target classes, such as nuclear hormone receptors or kinases, and these can be used for even broader specificity analyses. Now that we have our lead compounds identified, we can progress to preclinical validation in our sample workflow. In vitro safety pharmacology profiling typically includes panels of biological targets that show strong correlations with both preclinical and clinical adverse events. Many of the biological targets in current standard safety profiling panels are based on those compiled by Bose et al. In 2012 and Lynch et al. In 2017. Adoption of these standards by the drug discovery industry to test for liability has likely contributed to substantial drops in adverse events reported in NDA filings within the last decade. For example, testing three core cardiac ion channels, ERG, CAV1.2, and NAV1.5, as recommended by comprehensive in vitro pro-arrhythmia assay initiative, has improved the accuracy of pro-arrhythmia risk assessment. In 2024, the IQ-DRU Safe Consortium recommended additional previously underrepresented targets to improve outcomes further. Eurofins offers a variety of safety panels in both functional and biochemical formats that cover a large number of known liabilities and include these newly recommended targets. And these can be used at various stages of the drug discovery process, providing a complete in vitro approach to lowering the safety risk of a chronic drug. To demonstrate the potential utility of these in vitro safety pharmacology panels, we have some data with the PIM kinase inhibitor SGI-1776. This is a compound that had development discontinued after a phase 1 trial back in 2010 due to cardiac liabilities. We tested this compound in dose response curve format in the functional cell-based safety scan 47 panel, which is shown in the radar plot at right, where we have EC50 or IC50 values plotted. This panel identified micromolar activity at the ERG channel among some other targets that could have informed some of the risks prior to taking the drug to clinic. De-risking compounds with in vitro safety pharmacology can save wasted time and money on development of compounds with adverse properties. We also tested our three GLP-1RA peptides from our workflow as dose response curves starting at 10 micromolar in this safety scan 47 panel. We found that these compounds are completely inactive in all targets in the panel, which is in agreement with their generally favorable risk profiles. I'll note here that safety liability panels are target-based, which means they can rule out many secondary adverse effects at known targets for a given drug, but they don't necessarily identify systemic liabilities. What I'll talk about next is how we can model potential systemic liabilities to increase confidence using biological modeling in the BioMap platform. The BioMap platform consists of three important components. The models, or BioMap systems, are cultures or co-cultures of human primary cells and protein biomarker endpoints, modeling broadly different aspects of human tissue and disease biology. The second important component is a large reference database, currently consisting of biomarker profiles of more than 4,500 substances. Now, these include standard of care drugs, but also clinical stage candidates and even failed ones. Included are also experimental compounds, tool compounds, mixtures, and an increasing number of biologics, such as antibodies, proteins, and peptides. The database is used to provide deeper insights for new molecules, as we have developed a collection of predictive analytical tools. For example, we can benchmark against a standard of care drug from the database, but we can also perform similarity searches against the whole database. Another important aspect here is safety and tox assessment that can be performed based on clinical stage compounds in the database with known adverse effects. On the slide now, we have diversity plus profiles for semaglutide and tirzepatide at multiple physiologically relevant concentrations. Plotted are these response profiles across all 12 systems and all 148 biomarkers measured in this panel. The biomarkers are listed on the x-axis and the responses are normalized to vehicle control. So we plot decreases and increases of biomarkers in comparison to the vehicle control for this given run. We also always factor in historical vehicle controls, which are depicted here as the gray envelope in the background. Differentiating biomarkers that are fulfilling our significance criteria would be annotated and interpreted. In this example of semaglutide, we see a dose-dependent reduction in vascular endothelial cell proliferation, which is indicated by the thick gray arrow. And this was not observed with tirzepatide. There are also some indications of modest lowering of inflammatory, such as IL-8 and soluble TNF-alpha, and immunomodulatory, such as soluble IL-2 and soluble IL-6 activities, as well as changes in tissue remodeling with collagen 3. These could be related to the vascular protective effects that have been observed for semaglutide in patients. Overall, BioMap profiling indicated few issues with both these peptides, which is consistent, again, with the relatively safe clinical observations that have been seen. Biomap is a very powerful tool to evaluate novel substances and even combination effects. It is important to note that Diversity Plus is not always well suited for testing efficacy of drugs for some diseases, for example, in cases where the biology is not directly modeled. Particular areas represented include immunology, inflammation, fibrosis, dermal and respiratory, oncology, and cardiovascular biology. For other areas, the tool is predominantly used for safety de-risking and mechanistic insights. To demonstrate the utility of Biomap, I'll share this example from Pfizer that nicely describes how Biomap and the tox signature analysis can prevent late-stage attrition. Pfizer developed a CNS drug candidate, which failed in NHP studies. These compounds look safe both in vitro and in rodent studies, but the project was stopped due to allergic skin reactions observed in the monkeys. When Biomap Diversity Plus profiling and the in silico toxicity signature analysis were performed, we were able to deconvolute the mechanism of the observed toxicity and also show that it was off-target related. This demonstrates how applying Biomap early in drugs discovery can prevent such late-stage failures and is a beneficial final step in our in vitro anti-obesity workflow. To summarize, I've shared a set of in vitro assays that can be used to progress from target validation with the Obesity Light panel to potency determination in multiple assay formats, followed by in-depth mechanism of action studies with GPCR bias and orthologous assays, and broad specificity testing with the GPCR Max panel. In the preclinical stage, in vitro safety pharmacology testing can be performed to limit the potential for adverse effects and de-risk assets, and Biomap biological modeling can evaluate toxicity and more broadly systemic activity. Lead compounds that are identified from this workflow could then progress to in vivo studies. While not discussed directly today, Eurofins discovery can also support these studies, which can confirm compound efficacy in vivo obesity models. To look at downstream impact, they can determine if modulating targets, such as GLP-1, impact obesity in a translatable way, and then look at general and specific toxicity and tolerability measures, such as modeling nausea in the laboratory rat. Together, Eurofins Discovery offers end-to-end metabolic disease services, which can be integrated and managed with our Discovery One team of experienced scientific project leaders and program managers. Today, we focus specifically on obesity, but we can support a variety of metabolic programs for diabetes and other therapeutic areas as well. We have significant chemistry, biotherapeutics capabilities, and can work with a range of modalities. We have thousands of ready-to-use primary and orthogonal in vitro pharmacology assays, some of which were discussed today. We have translational cellular assays to support your metabolic disease programs, including Biomap and custom assay capabilities using relevant primary cells and patient-derived material. And finally, we support both in vitro and in vivo safety pharmacology with animal models of various metabolic diseases. To summarize some take-home messaging from what I have shared today, the obesity drug discovery landscape is evolving with emerging biological targets, therapeutic modalities, and safety considerations. In vitro discovery approaches can help accelerate and de-risk obesity therapeutic development by addressing complex mechanisms from target validation and hit finding to safety pharmacology. These include things like mechanism of action, such as identifying G-protein bias agonists with signaling that promotes compound activity. This is similar to tirzepatide. Orthologous assays to identify species specificity that can inform in vivo studies and ensure use of proper models, such as avoiding rodent studies with orforglipron. And also use safety pharmacology panels and biological models to identify liabilities earlier in discovery and reduce failures in the clinic. Eurofins Discovery is an all-in-one integrated drug discovery service provider from in vitro to in vivo, and we have the expertise and know-how to drive your obesity integrated drug discovery program. Finally, Eurofins Discover X, our products provider, offers cyclic AMP, beta-arrestin, and internalization assays, ready-to-use kits, and QC bioassays to support and enable every stage of obesity drug discovery programs using small molecules or biologics. Please visit our website for more information on our solutions for metabolic disorders. On there we have posters, flyers, white papers, and a really nice resource guide available. And you can also connect with an expert to guide your drug discovery needs. With that, I will thank you all for your attention today, and I would be happy to take any questions you might have.
Thank you, Dr. Oostdyk, for that fantastic discussion. Now, as I mentioned, we do have our third poll question, which should be coming up on your screens very shortly. And that question is, are you currently using or exploring integrated drug discovery services? Yes, actively, considering it, or not yet? And just one more time, are you currently using or exploring integrated drug discovery services? Yes, actively, considering it, or not yet? Great, thank you for all your responses. Now we'll move on to the final part of today's session, which is the Q&A session. A quick reminder that you can still submit your questions using the questions panel in the menu on the right-hand side. But I think we will get started with the first question. So, Dr. Oostdyk, you mentioned the complexity of obesity as a multifactorial disease. How do you see this influencing target selection strategies in early discovery?
Thanks, Sam, and thanks for the question.
No
I
problem.
think there is kind of, as I talked about, a lot of different aspects to obesity as a disease, and that means there are a lot of targets in there. So we know kind of the main classes of drugs right now that are effective are these GLP-1s, but with the rise of these combination therapies, there are a lot of other targets that can be included in our therapies as well. And at the early stage, that means we can look at kind of either drugs like retatrutide or something that is hitting multiple receptors in one, or we can look at these kind of combination therapies where we're developing both potent agonists for GLP-1, but also maybe GIP or amylins and things like that that can lead to hopefully better outcomes in the clinic as we get down the road there.
Great, thank you for that. And the next question I've got here is GPCR bias seems to play a significant role in metabolic therapeutics. Can your assays measure bias signaling?
Yeah, as I shared in the slides here, we can measure bias signaling. I had that example with GLP-1 and kind of the different assays, and then we can also pretty much use any of the different cell-based assays that we have and measure bias between the different pathways that can help develop compounds that have that beneficial activity, typically biased toward the second messenger pathways as opposed to limiting that beta-arrestin signaling that can lead to premature internalization and kind of reduce the activity of compounds. So yes, we can definitely do that with the different assays that we have.
Great. And the next question I have is, how do you determine which obesity model is appropriate for a given therapeutic hypothesis or drug target?
Yeah, there are a couple of different aspects there. Kind of at the target-based level, you're looking at the specific targets that you want. These early stage assays are not necessarily looking at the overall model, but as you get later in that pathway, there are a couple different things that can impact that. With orforglipron problem, for example, you need to make sure that the species that you're looking at has the correct activity for the compounds that you're working with. And then downstream from that, it's kind of a little bit related to the target itself and whether you're modeling the correct kind of human response there, or if you're looking at it in a orthologous species, that can play a role there as well. And then bringing in things like biomap and different modeling that we can do kind of without getting all the way into full animal studies is helpful, and also kind of addresses some of these novel alternative methodologies that the FDA is promoting these days and kind of limiting the actual in vivo studies.
Great. And the next question is, how do Eurofins Discovery's capabilities support the evaluation of combination therapies like GLP-1 agonists and other metabolic targets?
Yeah, again, I think that is kind of related to a lot of what I shared in the talk here today, the fact that we have many different target-based assays for thousands of different targets, pretty much covering any type of target that can be addressed at the moment in obesity. So I talked about kind of the ones that are looking at the incretins and those that are related and kind of focused on GPCRs, but other targets that are of interest are also things that we can address as well as people look to kind of target not just these base GLP-1s, but also newer ones as well.
Thank you. And what are some of the safety considerations that need to be factored in early during obesity drug discovery?
Yeah, the target-based safety panels that we offer are really easy and kind of proven way, so to speak, to address the toxicities that are at known targets. That's what we can do for target-based therapies. This kind of makes sure that we avoid cardiotoxicity, CNS toxicity, and those kind of things. And then kind of downstream, you can use Biomap for the kind of a little bit more systemic toxicity, especially with the fact that these anti-obesity therapies a lot of times have things related to nausea and kind of gut motility issues that can be addressed a little bit at the target-based level, but then also we can transition into in vivo models, which I didn't talk about directly, but we can look at as well, where you can model nausea, for example, directly in a rodent and kind of see how that safety liability is impacted as well. And that can be implemented at different stages during your drug discovery program, either with kind of an early on lead compound or something that you've developed down the road. But kind of moving that at an earlier stage in your discovery process is going to help limit development of compounds that do potentially have these just general safety liabilities.
Great, thank you. And the next question is, how firm is the arrestin/CAMP bias story now, years on?
How firm? Yeah, it's still developing. I think there is definitely more literature, more studies have been published that seems to promote the fact that that second messenger signaling is more beneficial. In opioids, I think it's been a little bit more, I guess, firmed up, to use the same word there, where that beta-arrestin signaling is responsible for a lot of the negative side effects that we have, and the second messenger signaling promotes kind of just the analgesic effects. There have been a couple of recent studies for GLP-1, for example, where we have the same kind of process with the pathways that are impacted by GLP-1. So by promoting that extended signaling with the second messenger and limiting beta-arrestin, it is definitely seen as beneficial. Whether that translates all the way into patients is still kind of, I think, being determined with trials and things like that. So it's still a developing area of research.
Right. And the next question is, how do you think about translation from in vitro, to human in terms of tissue sensitivity? And they've given an example, for instance, a partial agonist in humans, because of lower target tissue sensitivity, could be a full agonist in the sensitive CAMP assay, which could confound the interpretation of bias at the in vitro stage.
Yeah, that's a really good question, and that's where really kind of expression level is going to play a big role here. So in like our primary CMP assays and things like that, we have pretty high expression in these, so you get a benefit of having really good potency that you can see early on, but that may not translate, as the question said, directly into kind of tissue specific levels, and that's going to be a little bit of a maybe something that you can address by changing expression levels of the initial screening that you're doing. There have been some studies that show kind of high, medium, and low expressing cells do have different levels of response, both for Emax and for potency of some of these different receptors. And so that can be tied in at this early stage, where you can look at different expression levels, because that will confound the downstream response if you're just looking at kind of the full versus partial agonism. What you can use these earlier stage assays for is kind of ranking the potencies of different early stage compounds that you have, and kind of take that into more mechanistic studies later on with different expression levels for each of your receptors.
Lovely, thank you. And got another question here. Do you offer services for in vivo or ex vivo brain activity analysis in mice?
That is something that we'd had to talk to our kind of custom development team on. I believe that we do have a team that can work on in vivo studies like that. I'm not sure what they have off the shelf versus potentially custom studies that can be done there, but that's something where you connect with one of our experts. They can give you more details on that and help you kind of plan a study that may suit your needs.
Great. And the next question, how do you see AI or data-driven approaches influencing obesity drug discovery in the next 3 to 5 years?
Yeah, AI is definitely a hot buzzword right now, so to speak, and that's growing in the drug discovery industry as well as kind of in life in general. There's always a need for more data with AI to be able to feed these models, and kind of what we're seeing a little bit of a reduction in these large kind of HTS screens to an extent, where these modeling can kind of generate lead compounds quicker. But what that does mean is there's almost more need for a model and actual lab-based wet lab system to validate these models and feed more data back in. So there's a lot of need for this cell-based screening and things to make sure that what these AI models are putting out. So as AI helps us develop more compounds quicker, that can lead to more testing needed in the cell-based assay stage. And also potentially it'll, as we kind of go out a little more, couple of years or so, we may be able to identify more novel targets and things that maybe were not identified with our more traditional methods. And that can again lead to more testing needed in the lab to go along with that. But it's definitely a growing field that is very exciting.
Yeah, definitely. And I think with that last response, that's all we have time for. I just want to thank Dr. Oostdyk for a great discussion, great presentation, and thank you also everyone else for joining us online and submitting all your questions too. As you do leave the webinar, a short survey will appear on your screen asking you to rate your experience. If you're unable to complete it now, it will be sent to you by email shortly, and we would really greatly appreciate your feedback. The webinar will also be available to watch on demand. So on behalf of Drug Target Review and Eurofins Discovery, thank you all for attending, and we hope to see you again soon. Goodbye.
Thanks, Sam. Thanks, everyone.
Thank you very much.